Okay, this is a presentation on basic cytogenetics. So the objectives of this lecture include introduction to clinical cytogenetics, differentiating between reciprocal and Robertsonian translocations, recognize structural chromosome abnormalities, including deletions, insertions, and translocations, describe the difference between inherited and acquired cytogenetic abnormalities, and explain the concept of non-disjunction. Finally, recognize methods of cytogenetic nomenclature, including whole chromosome aneuploidy as well as partial chromosome deletions. So we started being able to look at human chromosomes in 1959. Humans have 46 chromosomes. We have 23 pairs. Most of them look exactly the same, except for the sex chromosomes we will talk about. What chromosomes actually are is kind of like a scaffold, a very tightly wound DNA that winds around each other, winds around histone proteins, and makes the scaffold of the DNA. So there can be inherited chromosomal problems, anomalies, which is what you inherit from your, when the egg and sperm get together. And there's also acquired chromosomal abnormalities. We're going to be primarily talking about inherited in this presentation, but when we get into hematology, we talk about leukemia, we will be talking about acquired chromosomal abnormalities, things that just happen in a few cells and may cause advantageous, growth of a cell in cancer. But we're going to be talking primarily about what happened in the egg or sperm that made that individual. So why would you do cytogenetic analysis? And one is prenatal, to find out if a fetus may have a problem. Advanced maternal age increases the risk for a chromosomal aneuploidy. Screening, we're going to talk about in the prenatal lecture, screening tests, and anomaly noted on the fetus. And to be honest, today, the number one reason for an amnio is that we see multiple anomalies in the fetus. Another thing is multiple pregnancy losses. We like to test the embryo or the fetus, but sometimes we also need to test mom and dad. And then a family history of mental retardation, birth defects, or a chromosomal abnormality. In the pediatric realm, developmental delay. I'll talk many times. I hate this word, but it's what we cause, because parents will say to me, is the child going to catch up? And the answer is usually not. Multiple birth defects, extra fingers, extra toes, an ear anomaly, things like that. And then finally cancer, which we're not going to talk about today, but it's probably the number one reason that we do chromosomes nowadays, which are, again, acquired chromosomal abnormalities. So tissue, basically anything with a nucleus. So in blood cells, T lymphocytes stimulated with PHA. Skin or muscle, we have to culture these. Placenta. So we're going to talk about this a little bit, but when the egg and sperm get together, divide, and it's 16 cells, 3 cells become the embryo. The placenta are those cells, the other 13, that make the placenta, that are from the conception. Products of conception, when someone has a pregnancy loss, whatever is excreted, and that can include placenta, fetus, whatnot. Tumors, we've started being better at culturing tumors. Amniotic fluid is basically skin cells in the baby that slough off into that fluid, and that's what we pull off to culture. Chorionic villi is basically a piece of the placenta. We pull off a piece of the placenta to culture. So this is a chromosome spread, and it's banded, but you can see here are 2 cells that didn't pop open. So here are our chromosomes, and they're kind of random. So here's a 21, here's a couple of 22s, here's one of the chromosome 1s, and here's the other one upside down. And over time, you learn how to analyze these. And when you analyze them, you put them in size order. So a karyotype is a chromosomes put in size order. The largest human chromosome is 1. The smallest is actually 21, but they were working with unbanded chromosomes when they diagnosed Down syndrome, so they thought 21 was smaller than 22, but by the time they figured that out, Down syndrome and trisomy 21 was synonymous, so they never switched it back. But the other important concept here is that the size of the chromosome doesn't tell us how much genetic material is there. The chromosome with the most genetic material is 1, followed actually by X, 2, 3, and then, everyone would guess this, 19. So this is a very, very gene-rich chromosome. The chromosome with the least amount of genetic information besides the Y chromosome is 21, which makes sense that this is the most likely to result in a live born or the with Down syndrome. And then other chromosomes with not as much genetic material include 13 and 18. So it actually makes a lot of sense. But again, most of the chromosome is non-coding, although I'm sure it's important stuff. I just don't know what it is now. And here we have the male. Again, this is it has a number of genes, but a lot of repetitive area. The Y chromosome determines male sex. So let's talk a little bit about the anatomy of the chromosome. So the middle of the chromosome is known as the centromere, and this is where the spindle fibers attach to. This is repetitive DNA right here. The short arm is known as the P arm. A lot of the early nomenclature was done in France, and P stands for petite, and Q is the next letter in the alphabet. So the short arm is the P arm, the Q arm is a long arm. The ends of the chromosomes are known as telomeres. And there's another thing, when they talk about telomeres, they seem to get shorter every time cells divide, and that has to do with aging, although we're not 100% certain all the things involved in that. But anyway, if there's an additional chromosome, well, instead of 46 chromosomes, we have 47, XX or XY, and then which one is extra. So this is the nomenclature for an extra chromosome. If there is a deletion of part of a chromosome, deletion and which chromosome, 5, and then where is it? P14.2. P14.2. When there's one number, everything above here is missing. If it is an interstitial deletion, a deletion and then the chromosome reanneals, let's see, deletion, here we have chromosome number 5, P11.2, P12.3, 11.2 to 12.3. So this piece right here is missing. That would be the nomenclature for an interstitial deletion. So if you see two numbers, that means that it's interstitial. 1, it means it's terminal. Everything, behind this, 1.4, anything over here is all missing. Now, the same nomenclature that we used when doing cytogenetics, we also use for locating genes, and that has to do with linkage analysis and all kinds of things like that. So here we have the long arm of chromosome 7, where the gene for cystic fibrosis, or the CFTR gene, is located. A transcript is made, a protein is created, and this protein sits in the cell membrane to transport chloride, which we will be talking about in the future, but the CF gene is located 7q31.3. You do not need to memorize these locations. I'm showing you this as an example. So aneuploidy is the most common cause of pregnancy loss. Of all pregnancy losses in the first trimester, half have a chromosomal abnormality. Women over the age of 50, a fetus of a woman over the age of 50, 1 in 50 have a chromosomal abnormality, but by birth, it's 1 in 160. Numerical, in pregnancy losses, 96% are extra or missing chromosomes, 4% are unbalanced rearrangements. Women over the age of 35, the vast majority, again, are numerical, with 15% to be structural abnormalities, 10% are balanced, which we're going to talk about in a second, and 5% are unbalanced. And by the time you get to birth, 60% are numerical, and here we have balanced and unbalanced, and we'll talk about how balanced aren't even a problem usually. So of this 1 in 160, only 70% of them, 60 plus 10, would result in a baby with an issue. So you can see as you go through the pregnancy, the risk of a chromosomal abnormality decreases, and that's because nature, if the baby has a severe aneuploidy, the pregnancy does not continue. So chromosomal aneuploidies you need to know include trisomy 21, trisomy 18, trisomy 13, 45X, 47XXY, 4P deletion, and 5P deletion. We are going to be talking about the specific clinical phenotype of this in a future lecture, but these are the ones you need to know. So during meiosis, it's a reduction division. We go from 46 to 23. So mom gives half of her chromosomes, dad gives half of his chromosomes, 23 in the egg, 23 in the sperm, and the fetus ends up with 46 chromosomes. And that's how things usually go. But as with anything, mistakes can happen. So this is just an example of two chromosomes, the blue chromosome and the gray chromosome. They divide evenly. One parent gives one blue and one gray. In this situation, there's non-disjunction at the first meiotic division that both of the blue chromosomes go to one side, and then in the second, and then we separate. So at the end of the day, one parent gives two blues and one gray, and the other just gives a single gray. If this is used for fertilization, 1, 2, 3 copies of the blue chromosome and 2 copies of the gray chromosome, and in here we only have a single contribution of the blue chromosome. Monosomy is almost always lethal. The only exception to that is Turner syndrome. Trisomy is usually lethal, and the exception to that is plus 21, plus 13, plus 18 as far as resulting in a live-born. So as I showed you a little bit earlier, products of conception is defined as whatever is expelled from the uterus during a pregnancy loss. When we look at the chromosome material of early pregnancy losses less than 10 to 12 weeks, 50% have abnormal chromosomes. Chromosome analysis should be considered on a woman who's had 2 to 3 early pregnancy losses or an older fetus that was lost with multiple anomalies. And that's actually looking at the mom and dad. Separation of the fetal tissue when we're looking at fetal tissue is really important. So, for instance, when I run my lab for products of conception, if 100% are normal female, then I probably did not separate the tissue, and that's actually one of my quality controls. So, I should expect 50% of my products of conception to be male and 50% to be female. And if that's not what's happening, then I'm not doing it right. Down syndrome, we're going to talk about the clinical characteristics of this in clinical cytogenetics, but basically all the individuals with Down syndrome have an extra copy of 21. Most of the time, it's because of non-disjunction. So 46XX or XY plus 21 is seen in 90 to 95% of individuals with Down syndrome. 2 to 4%, the conception started with 47XX or XY plus 21, but in a group of cells, this extra 21 was lost. So we do have some cells that are normal. The higher the amount of the normal cells, the more mild the phenotype. But it's also, all the cells are different, so it might depend on the organ where it has a higher percentage of mosaicism. And finally, a translocation occurs in about 2 to 4%. So if you have an individual with Down syndrome, 95% of the time they're going to look like this. 2 to 4% they can have mosaicism. I'm going to talk about a translocation in the next slide, but the only one of these that would result in a milder phenotype is mosaicism. So a Robertsonian translocation is a translocation between two acrocentric chromosomes. Acrocentric chromosomes are 13, 14, 15, 21, and 22, that the short arms are a repetitive material, and probably because they are repetitive material, they seem to associate together in the interphase nucleus, and translocations can occur. So this individual has 46 chromosomes, and I told you 46 is normal, but there's a derivative chromosome, 1421. So this chromosome is a 14 with a 21 on top of it, plus they have 2 more 21. So 1, 2, 3 copies of 21. The loss of this repetitive material during this translocation is really, it's repetitive. There's 10 copies versus 8 copies. It really doesn't matter. But so an individual with Down syndrome would have 46 chromosomes, but they would have a derivative chromosome. That means this chromosome right here. And then at the end, you see plus 21. A carrier of a Robertsonian translocation, and approximately 1% of the population carries a translocation, would have only 45 chromosomes. And this derivative chromosome would, so it's 1, 2 copies of 21. Now this individual is fine, but when we undergo meiosis and there has to be synapsis, that's when we get into trouble. So when the chromosomes are trying to line up for crossover, they have to line up in this kind of unusual tetrad. Now, if the 1421 migrates to one cell and the 2114 migrates to the other in the alternate segregation, then when it's fertilized, we get 2 14s, 2 21s, and they're all in the right place. In this one, we get 2 21s, 2 14s, even though they still have a Robertsonian translocation, normal phenotype, normal phenotype. But if the 1421 migrates with the 21, leaving the 14 alone in the other cell, or the 1421 migrates with 14, leaving the 21 alone, when this is fertilized with normal gamete, we get 2 14s and 3 21s. This is Down syndrome. When this is fertilized, we have 2 14s and 1 21, 3 14s and 2 21s, and 1 14 and 2 21s. This is lethal. This is lethal. This is lethal. This is the only one of the from the adjacent that results in a live-born. And as a result, when a parent is a carrier of a balanced translocation, there's a 15% chance of having a child with Down syndrome. You know, it's like, well, it's like there's all these possibilities, or 6 possibilities, but the vast majority are here, and all these are lethal. So here's just a diagram. Here's our normal segregation, alternate segregation. The 1421 goes together, and the normal 14 and 21 go together, and this would result in a normal fetus. And adjacent, here's adjacent 1. Here we have the 21 going with the 1421, leaving the 14 alone. So, again, if a parent is a carrier for a 1421 translocation, there's a 5 to 15% chance of having a baby with Down syndrome. If the baby is born with Down syndrome and has a 1421 translocation, there's only a 50% chance that one of the parents carries it. It could have occurred in the sperm that made that child. So when we see a baby with a 1421 translocation, we first have to do the parent's chromosomes before we run to the conclusion that they're carriers. Now that is a special translocation, Robertsonian, in between 2 acrocentric chromosomes. The next thing I want to talk about are reciprocal translocations. And reciprocal translocations are translocations between any 2 chromosomes. So let's call this chromosome 1 and this is chromosome 2. So in the parent with a reciprocal translocation, they have a normal 1 and a 1 with sum of 2 on it, a normal 2 and a 2 with sum of 1 on it. When you add it all together, they got 100%. The chromosome is considered, the name of the chromosome is considered by which one has a centromere. So sometimes there'll be a really large piece of, let's say, chromosome 1 at the bottom of 21. There's more material from 1 than 21, but it's considered chromosome 21 because the centromere is from chromosome 21. So what happens when this is when we undergo meiosis? Well, the 1 could go with the 2, and when that's fertilized with a normal parent, everything's great. Or the 1 with part of 2 on it goes with the 2 with part of 1 on it. And here we have a normal 1, a normal 2, 1 with part of 2 in it, 2 with part of 1 on it. When we add it all together, we're fine. The third possibility is 1 migrates with the 2 with part of 1 on it. And when this is fertilized, we have extra 1 and missing 2, and then finally 1 with the normal 1 with 2 with part of 1 on it, and we have extra 2 and missing 1. And these are known as duplication deletions. It kind of depends on how large the piece is. What's interesting is the larger the translocation, the more likely, if you actually had a pregnancy that made it, it's either this or this. Small translocations can result in liveborns with multiple anomalies. So, you know, sometimes we'll see a family and there's actually calculations we can make, which you do not need to do, to determine what their risk would be. Okay, so translocations, as I said, can occur between any 2 chromosomes, and while an individual with a balanced rearrangement is normal, there is a risk for the chromosomes not to separate during meiosis correctly. It's also possible that a gene can be affected during the translocation. So when a translocation is de novo or new in that individual, it could be the cause of their issues. When there's an imbalance of genetic material, there's a risk for either pregnancy loss or the baby with multiple anomalies. So when it's an unbalanced rearrangement, there could either be a very high risk of pregnancy loss or congenital anomalies. Approximately 1% of the population carries a translocation between 2 chromosomes, and a reciprocal translocation can, or either reciprocal, either Robertsonian or just reciprocal, can occur from mom or dad. So when we have a couple with multiple pregnancy losses, we need to look at the chromosomes of mom and dad. It's not just mom. So a USMLE-like question: a baby has had chromosome analysis. The results indicate 46XY translocate 1421 plus 21. What is the diagnosis? So there is a Y chromosome that makes you a male, and there's 46 chromosomes, but there's a Robertsonian translocation, and this is indicating an extra copy of 21. So this is a male with Down syndrome. And that concludes this presentation.